In this video, we will discuss about the problem or disease that is galactosemia. It is a single gene disorder, and the gene that is mutated or not functioning properly is galactose 1 phosphate uridyl transferase, and this gene mainly work in the liver. So let us first understand what is the normal functioning that is related to galactose 1 phosphate uridyl transferase, or in short we can call it as GALT. GALT. So in the liver, uh, this beta galactase enters in the liver, and this beta galactase is converted into the alpha galactase with the help of enzyme GALM, and this alpha galactase will then be able to react with the enzymes such as GAL will then be able to react with the enzymes such as GAL, GALT, and GALE. So, however, in patients that are affected with galactosemia, this GAL is not functioning properly. Because of this, there is a accumulation of alpha galactose 1 phosphate observed within the patient, and within the first few weeks of the birth the patient start uh, getting symptoms patient starts getting symptoms that is related to galactosemia, and one of the hypotheses is that the cell is not being able to produce energy because of the enzyme that that becomes inhibit because of the excess of galactose 1 phosphate, and that enzyme is galactose 6 phosphate that helps in the conversion of glucose to energy. So one of the hypotheses is that because of the excess of this galactose 1 phosphate, the galactose 6 phosphate enzyme get inhibited, and thereby the energy production within the cells get reduced. And another problem that happens is that yeah another problem that happens is that because of the non-functioning of the GALT, this will also impact the glycosylation, the further glycosylation process. And when the glycosylation process get affected, or we can say the post-translation modification get affected, the proteins will not be able to function properly, as glycosylation is considered as one of the important uh, post-translation modification that is responsible for uh, proper functioning of the proteins. So in the absence or improper glycosylation, proteins will not be able to function. Most of the proteins will not be able to function properly, and this galactosemia at present, the one of the remedies of this, one of the treatment we can say, or prevention, yeah, one of the prevention of this galactosemia is the dietary inhibition, that is to restrict the diet that contains galactose. So that restriction might even include such as restriction of some fruits, vegetables, uh, and sometime even some essential uh, foods that are necessary to provide the nutrients to the patient. So sometimes this dietary restriction will leads to a lack of nutrients within the patient, and however, so far there is no direct cure that has been, no permanent or cure that has been that has been made against this galactosemia disease. So people has to uh, continuously suffer from the symptoms that occurs through because of galactosemia, and this function, as it is shown in the figure, is mainly occurs within the liver uh, when the beta galactose enters the liver. So uh, the so however, there is another hypothesis that has been there uh, that might cause other side effects because of the absence of galactose 1 phosphate uridyl transferred affecting the uh, energy requirement of the cells. However, the in vitro results against in vitro results of uh, this absence of galactose 1 phosphate uridyl transferred supports supports the hypothesis that uh, this excess of alpha galactose 1 phosphate will inhibit the galactose 6 phosphate and finally cause the energy deficient or energy insufficient condition within the cells, as galactose 6 phosphate will not be able to properly convert this glucose into the energy or ATP because of the inhibition of galactose 6 phosphate in presence of excess galactose 1 phosphate. If you need to understand this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.